So now let's talk about this issue with mutations. So mutations occur when DNA is different, or it's changed in some way from what it originally should be. So for example, if the DNA sequence is the G, C, C, T, A, G, C, etc., if all of a sudden the C gets converted to an A, now we have a mutation because the base that is supposed to be in this spot here has changed from a cytosine to an adenine. Now, this can happen for a variety of reasons. First of all, DNA gets replicated a lot, and it's just like if you're trying to maybe type up something from an original, and as you're typing, you hit the wrong key. So you just have a typo. So sometimes during DNA replication, or even during transcription, sometimes the wrong nucleotide just gets put in the wrong spot. Environmental factors can also damage your DNA. So things like exposure to radiation. So for example, UV radiation from the sun can damage the DNA in your skin, which leads to skin cancer. Or you could be exposed to certain chemicals. For example, some of the chemicals in cigarette smoke can lead to damage in the DNA in the lungs, which can lead to lung cancer. Now, if DNA changes, it means that our mRNA is also going to change, right? Because the mRNA is made based on the DNA template. So remember, G pairs with C, C pairs with G. And in our original, we would have a G here, and then the T pairs with A with U, G with C, and C with G. But if our DNA mutates before mRNA is made, now we're going to have the wrong mRNA nucleotide in that area. So we're going to have a U instead of a G. Now remember that the mRNA is what the ribosome uses to make a protein. So now we've changed the code, or we've changed the recipe for how the ribosome is going to make this protein. So ultimately, we're going to end up with a different protein that might be the wrong protein. But the good news is, if the DNA changes and the RNA changes, that doesn't always mean that the protein is going to change. And that's because if we look at our chart over here, so this is our codon chart for our amino acids, and you'll notice that multiple codons might end up with the same amino acid. So depending on where the mutation occurs and which nucleotide is involved and which nucleotide gets changed to which other nucleotide, sometimes it doesn't end up changing the end result of the protein. So for example, let's take a look at what we did up here. So our original codon was GCC. Oh, sorry, just kidding. Our original codon was CGG, and our new codon with the mutation would be CGU. So let's see which proteins are made, which amino acids are coded for by these codons. All right, so CGG codes for arginine, whereas CGU, ooh, surprise, it also codes for arginine. So in this example, the mutation didn't matter because we're still going to end up with this amino acid in that spot in our polypeptide. So no big deal. Now, if it were to, let's say instead of changing this third letter, we change the first letter. So now let's say that G gets changed to a C, which changes our mRNA, the first nucleotide, to a G. So now we've got GGU, and GGU, let's see, so GGU, that codes for glycine. So now we've got a problem because now instead of arginine, we have glycine in that spot, And now we have a protein that doesn't have the right polypeptide chain. It's got the wrong amino acid in the wrong spot. And that means that the secondary and tertiary structures of this polypeptide are not going to be correct because glycine has a different side chain than arginine does. And in doing so, it's going to make our protein fold in a different way, and it's potentially going to make our protein not function the way that it's supposed to. All right, so not all mutations cause a change in the protein. It really depends on which nucleotide is affected, the location of the nucleotide, and which codons code for which amino acids. Now, let's say that our mutation did change the protein that was made. Now, that's not always bad, but we'll start out by talking about some of the bad things and then move on to maybe talking about how sometimes it can actually be a good thing. But let's start out with the bad. All right, so if this mutation causes the protein to be made in a way that makes it lose function, or maybe it's a protein that regulates the cell cycle, and so now the cell cycle is not regulated properly, and it can lead to problems, especially things like cancer, and then most genetic diseases are based on a genetic mutation that leads to a protein not being made properly, and other disorders as well. All right, so these are going to be the bad possible outcomes that can lead to a lowered quality of life or even death. But remember how we said that some mutations are totally neutral because of that redundancy in the amino acid code. So if the mutation does not change which amino acid is located in which area, then it doesn't matter. And we call this a neutral or a silent mutation because you'll never know that you have it because it really doesn't matter. If you end up with the same amino acid code, even with the mutation, then it doesn't matter. It's kind of like with, let's say you've got a recipe for cupcakes, and in the recipe for cupcakes, you need to have eggs and sugar and maybe some water and oil and some flour. And then let's say that you accidentally change this recipe so that instead of using sugar, you're now going to use organic sugar. Are your cupcakes going to taste any different? The answer is no. It's sugar. Whether it's just regular sugar or organic sugar, the substitution doesn't matter. You're not going to notice a difference in the end. Now, if it were a bad mutation, it would maybe mean that the sugar, instead of adding sugar, you added salt. All right, now that would be a bad mutation. You're going to end up with cupcakes that don't taste very good. But if the substitution is basically the same thing, or if it is the same thing, then who cares? Now, last but not least, but sometimes mutations are actually beneficial. So sometimes they're actually good. So it might be the sort of thing where the incorrect protein is made, but instead of making a worse protein, you actually end up making a better protein. So let's take a look at these insects here. So let's say that this resistance refers to being resistant to insecticide, like a bug spray. So if you have these bugs in your house and you spray it with Raid or some sort of insecticide, what's going to happen is that the individuals that don't have this beneficial mutation are going to die. But the individual that has this beneficial mutation is going to survive. Now, if this individual then goes off to make a whole bunch of little baby bugs, now all of these little baby bugs have that resistance built into their genes, and so they're going to be more likely to survive if they're exposed to that insecticide. So it sucks for us as humans because we have to keep making stronger and different insecticides in order to kill off these insects that we don't want, but it's good for the insect because it means that they survive and can reproduce. And this is actually the basis for evolution. So if the mutation results in some sort of beneficial trait that increases survival and reproductive output, it means that more individuals in the future generations are going to have that beneficial mutation. And then, of course, I had to put in the Teenage Mutant Ninja Turtles over here. Obviously, they're not real, but the mutant part of their name indicates that they have a mutation that allows them to, fight off criminals and eat pizza and live in the sewer.